told you several times, recombination is a form of DNA repair. It's also happening in a very different context in meiosis. So I'm not talking about that in the context of both processes because it's the same biochemical mechanism. So after we finish that part, we're going to talk about what I would call gross chromosomal rearrangement. That's the next chapter, chapter 13. And we'll finish off with transposition. So I told you last time to make the to ligate the two lectures. To repair that break between last Tuesday and today. There were three ways to fix a double-strand break, and one way is non-homologous conjoining. I discussed that. I told you it was an error-prone process. And another way, which I finished on that last time, was the process shown here on your right, and it involves DNA synthesis, so it's called Strand Invasion DNA synthesis, so it's SDSA, and you'll have a double-stranded break, and that resects the 5' ends around that break, produce something about somewhere around 100 to 200 base overhangs, and the overhang strand invades into the unbroken template, and that, of course, can be the sister chromatid after DNA replication. It really can be an identical sequence somewhere else. It doesn't have to be sister chromatid. But this is a repair process that is effective after DNA replication in G2. There's local DNA synthesis, and past the point of the break, and you see that the newly made strand here can reanneal with the overhang that was produced, and DNA polymerase can fill in the gaps, and that gets ligated, and the repair is fixed. And this is a precise repair mechanism. So now we move on to recombination. So this is a little bit more involved. And first, I'd like to point something out that is rarely made clear. It's simply that oftentimes a confusion of terms between recombination and crossing over. And crossing over can be a product of recombination, but it is not always. Recombination does not always lead to crossing over. So what's a crossing over? It really depends on the final product. You can see here, if the final product is actually, if the strand is actually a true pairing or religation between two non-sister chromatids, you have a crossover. So you have one part of the chromosome is one origin, the other part has that's ligated to it as a result is of another origin. Okay, so that's a crossover. And that can be the result of recombination. But recombination does not always lead to that. It can lead to partial formation of recombinant molecules, but overall the chromosome stays at the same origin. And this is a strand exchange only, locally, as you can see, as I depicted here, very simple diagrams. And this is a non-crossover situation. So we're going to discuss on how that is produced. So the different steps in recombination, I outlined them here for you on the top of each slide. Step one is the production of a double-strand break. That's in meiosis. So meiotic recombination, in principle, these cells are not experiencing DNA damage. There is a specific activity that produces a double-stranded break. Okay, but if there is a double-strand break, say in vegetative cells and non-meiotic cells, due to gamma radiation, for example, or problem in DNA replication, then that is not a reduced DNA double-strand break. It's an accident, it's damage, and that in itself can induce recombination. So in other words, double-strand break is an intermediate, is an initiator of recombination. So the first step in meiosis, no damage, no chromatids are broken, is actually formation of the break. The second step, which you're going to see in later slides, is there is a strand invasion step and formation of a structure, which I'll show you, called the D loop. Step number 3, formation of heteroduplex on both chromatids, in fact, on both non-sister chromatids, and formation of a structure, which is very important to know of, is the structure called the double holiday junction. And in step number 4, there's resolution of this structure of the holiday junction and separation of the chromatids involved. So I told you in the case of the cells that are experiencing damage, they've already have, let's say, a double-strand break, and that's going to be repaired by the mechanism we're going to talk about now. In meiotic cells, there is a specific enzyme, which is an endonuclease, which will produce in a regulated manner this double-strand break. And this enzyme, this endonuclease, is called SPO11. And that's important to know and to remember. SPO11 is a meiotic specific enzyme. It is only expressed in meiotic cells. It is not even transcribed in cells not undergoing meiosis. So in vegetative cells, you can forget about SPO11. SPO11 is not even there. As a result of a meiotic program that cells are experiencing, say germ cells, they express SPO11, and this endonuclease is able to produce double-strand breaks, as shown here. These double-stranded breaks, importantly, are then on both sides of the break resected, and there's a specific strand that is being resected. It is the 5' end. And as you can see, this produces 3' overhangs. What I mean by 3' overhang is, I mean a piece of single-stranded DNA of something like 100 to 500 nucleotides that actually presents a 3' end. And you see both 5' ends are resected after the break, and in meiotic cells, the break produced by SPO11. The activities that are responsible for this resection have very recently been identified, and I'm not going to go into the details. All I want you to know is just a simple exonuclease that is going to resect the 5' end. It's a complex set of activities which together will lead to the formation of the overhang. If you want more detail, then we'll have to discuss that some other time. But the important point is the activities required for the formation of these overhangs is a complex called MRX and an exonuclease called XO1. I have this in a later slide, but I'll tell you now. MRX stands for MR11, RAD50, and XRS1 or NDS1. These are three subunits of that complex, which participates in the formation of the overhang and the resection of the 5' end. And so what we get from that is a molecule that's broken and that is exhibiting these hundreds of nucleotides on both sides of the break that are single-stranded and that present What happens next is step number 2, strand invasion. This is a process. First of all, let me describe what this is. Strand invasion is the invasion and the hybridization of the annealing of one of the overhangs, and that will, remember, has a 3' end into the duplex of the non-broken chromatin. This requires exquisite base pairing. So in these 100 nucleotides or so, there's a single mismatch that cannot work. It's not stable. And this is to say that the initiation of recombination requires a perfect sequence identity. This is why homologous is called homologous recombination. That's why it's called homologous, because it only works initially. The structure can only be formed when the two sequences, the sequence that's invading the non-broken chromosome, can base pair and are absolutely complementary, that is identical. So now, regarding this strand invasion, this has to be catalyzed. And the molecules that catalyze the strand invasion are also very important to remember. I will list all these things at the end for you, so you don't have to worry about it. It's on the slide. In bacteria, because this happens in bacteria too, it's called REC. In vegetative cells, non-meiotic, mitotic, that activity is called RAD51. And in meiotic cells, it's called DMC1. How they work is truly fascinating, and that is, we don't fully understand it yet. They are able to allow for a genome-wide search for sequences which can recombine with this area. And it is believed that these molecules, RAD51 or DMC1, essentially allow for a sampling and trial and error of hybridization between the overhang and a possible recombination template. But in any event, they're essential. If you are mutated, cells that are mutated for these components, see a big drop in recombination. It doesn't occur. This process doesn't occur effectively. The strand invasion process constitutes a structure or structural element in this region called the D loop. D as in displacement. And this is this blue unbroken piece here that essentially is identical to the overhang that's doing the strand invasion, and you see it no longer hybridizes to its strand in the double helix. It is displaced. And so the D loop, if I go back to this little figure here, if you look at my laser pointer, that's the D loop. Okay? So I don't know why they pointed over here. It should be here. Right? But it's logical, it's a loop. And so here we are in the situation of an invading strand catalyzed by RAD51 if you're in mitosis on the vegetative cell, DMC1 if you're a mitotic cell. And have an unhybridized, following that event, there is a little bit of DNA synthesis. This, after all, is a 3' end, remember? So it's going to be elongated by DNA polymerase. So DNA polymerase is going to elongate this using the strand at the bottom as a template. All is fine. And if you look at this, the D loop is simply complementary to the other overhang, right? Since the invading strand hybridized to this other strand before the break, logically, it must be that the top overhang here can hybridize to the D loop, because the two double pieces are identical. Watson and what Crick strand they can hybridize. And there will be a little bit of DNA synthesis and elongation of that 3' end as well. Now we're forming a structure that's a lot more stable suddenly, because the invading strand starts to have a lot more complementarity. Therefore, there's more energy to that association with the template, and the D loop as well then will associate and hybridize with the other overhang. This DNA synthesis will continue locally. We're talking about a few hundred base pairs here. And eventually, the extent of the DNA that was resected by MRXX01 is going to be resynthesized. It's going to be resynthesized using the non-broken strands as templates. Ligation is going to occur over here, and ligation is going to occur here. You see my laser pointer between this 3' end and this 5' end. That's the one that was resected before, to actually fully recreate complete unbroken strands. The structure that we've just built is called the double Holiday junction. Holiday of the name of the Yale scientist who first proposed this mechanism. And so now we have double Holiday junctions. We have ligated the strands, as I told you, and you will note that we have local formation of heteroduplex regions on both duplexes over here. That's the part that first hybridized with the D loop, and over here, that's the part that was first extended using the 3' end of the invading strand. So we have heteroduplex formation on both chromatids. I'm showing here meiotic recombination again using a non-sister, but the same thing is happening for a DNA break that's being repaired. We are left with this, which of course now we need to resolve. Okay, we need to liberate those DNA strands from each other. So this structure needs to be resolved, and it can be resolved in two ways. And this is what decides whether you get a crossover situation as a result, or a non-crossover situation, as I showed you a few slides ago. The way the double Holiday junction is resolved is, and this is not the first time I tell you this and not the last, it is not known. So it's the subject of intense biochemical investigation, but we have yet to discover here the enzyme, the resolvase, that will lead from the double Holiday junction to fully separated strands which can go their own way. Okay, so what I'm going to tell you now is going to be, in fact, a model, but we don't have any molecular details on this. this double holiday junction can be resolved in two ways. The first way is very easy to picture. You see, we have redrawn this. You can simply, essentially, cut the strands and religate them horizontally. And you create this structure over here on both sides where the strands are crossing each other. So this is called east-west resolution on both sides. And if you do an east-west resolution, what you'll see, in fact, is look at these chromosomes that you get back. Red, heteroduplex, red. That's a non-crossover situation. Same thing for the bottom one, is a blue chromosome with a heteroduplex. So if you do east-west resolution of the holiday junction, that yields a non-crossover situation with some degree of heteroduplex due to the formation of the holiday junction. But you can also resolve that another way. There are other ways to resolve this that we're skipping. But the point is you can resolve one of the junction in a configuration called north-south, when in fact the template strands are cut and religated. So you cut over here, and this blue piece here can religate to the top one and vice versa. That's also something that is detected. And what you see here is, as a result, you see red chromatid, heteroduplex, blue chromatid, and the mirror image there below. And this, of course, is a crossover situation. So what is actually observed experimentally is 50-50. So double holiday junction will yield 50% of crossovers and 50% of non-crossover configurations. This resolution from a biochemical standpoint is impeccable. That is, the polarities of the strands are conserved and respected, and this is actually achieved to the nucleotide. So this is an exquisitely precise repair system. And of course, in meiosis, we're not losing or gaining any nucleotides. So if this is used as a repair system, it is a precise repair system, and once again, this requires a non-sister as a template, non-sister chromatids, for example, in G2, but it can also happen in G1 to the extent that if the break occurs in a repeated sequence or a sequence that's present on the chromosome, recombination can also occur here. Remember mitotic recombination? Same process. Questions? I think there was a question here.
I'm confused what the heteroduplex region is.
Well, I'm gonna. So, where does the heteroduplex come from? So I'm going to go back to I think step 2. So the heteroduplex. Maybe you look at this. So you understand there is strand invasion. Just look at this strand, right? And what's going to happen next, in fact, this pairing here really doesn't fix anything, doesn't fix the break, because by definition, the 3' end represents the site where the DNA is broken. So in order to, in fact, be able to repair this, there is local DNA synthesis right here. See this dark red piece? We can use the unbroken chromatid as a template. DNA replication will occur and go past the break, such that we can religate the strands and not lose any genetic information. But you see that this, we have a red strand, a red chromosome, or chromatid, I should say, hybridizing with a blue chromatid. That's a heteroduplex. That's what we mean by heteroduplex. And same thing over here, all right? I just gave you a happy answer. Over here, you're also making a heteroduplex. Just, refilling the gap. Any other questions? Great. Yes?
I'm not sure. Could you explain, like, just clarify it a little more? So how does that holiday junction, that intersection between the incon and the decon, how does that form? Is there some sort of like?
So the holiday junction forms after ligation, and I'm going to show you where, because this is the holiday junction. So if you look at this, okay, let's look at this slide here. You have a nick after synthesis, right? You have a nick over here. You have this 3' end being elongated. There's a 5' end here, and there's a nick religation over here, first of all. And there's another nick over here. So you have the 3' end was elongated and the 5' end here. And if you look at my little pointer, this 5' end and this 3' end are going to be religated as well. So we have two points on the DNA molecule that need to be religated. All right. So remember this, crossovers don't cross over. We don't fully understand it, but essentially it's the way the holiday function is resolved, and you can get 50% of the time a crossover recombination product and 50% of the time a non-crossover. Now, we have a little bit of a problem. And that problem is, I told you that the recombination really needed very precise sequence identity, right? Because the base pairing is essential for that, the Strand Invasion. But then at the same time, I'm telling you that recombination can actually generate genetic diversity, right? In meiosis, I've been on and on about this, right? Told you, you can recombine between alleles and form double mutant or, things like that. So there's a little bit of a conundrum. How can this happen? Well, let me first answer that question, and then I'll tell you what happens when there are sequence differences. The conundrum is that really, it's fairly easy to resolve actually. You can recombine between vastly different sequences. Okay, that is, a template can be very different from the broken strand, for example. But it is initiation of recombination that requires very precise sequence identity. And once the holiday junction has formed, it actually is not necessarily a solid structure that doesn't change. There is a process called branch migration. And branch migration, if we go back to a holiday junction, is a process by which the strands can essentially de-anneal from each other and re-anneal. So in other words, the amount of heteroduplex can greatly increase by moving these junctions here on both sides. So this one towards your left and this one towards your right. These are points where the strands can separate. So essentially the blue duplex here can separate, and the bottom strand can re-anneal to red strand, chromatin, and then form extensive regions of heteroduplex. And that doesn't require necessarily very precise sequence identity. So this is where you can actually integrate in the segment that's recombining huge chunks of DNA that are not identical. Okay, is that clear? Yeah.
I just have a question about the previous slide. You said the 5N will be reliking it, and what else? What other N will be reliking?
What are the 2 points that are being relocated?
Yeah.
So I'm going to go back to this. So if we look at this this diagram, so over here there's a 3' end being relocated with the 5' end that's resected, and over there. So this 5' end not really shown. I mean, you see a gap, but in reality you can see them close to each other, and there's this 5' end being relocated to the 3' end. In other words, the 5' ends that were recreated by resection need to be resealed. Right? Okay, so do you get this point though about branch migration? That what the holiday junction is really the initiation of the exchange, but the amount of DNA that is participating in the exchange can actually represent a lot more than this little piece that I just showed you because of branch migration. And therefore, we can then integrate by branch migration mismatches, deletions, allele mutations, and things like that that are not absolutely identical between the 2 strands. And so what happens then? The cool thing is we've already discussed that. So if you have, say, a sequence different, for example, you have a segment here that is a CG in one of the chromatids and AT in the other chromatids, you might get that situation in the heteroduplex. And that's a mismatch. And that is dealt with by the mismatch repair machinery. So mut-S, mut-L, mut-H. And in mammals and in eukaryotes, essentially this is done randomly. So you have situations where, in fact, say the red strand here is going to be changed to the blue sequence. So we're going to get a transversion AT. And if we look at yeast, but it doesn't have to be, you get a 3-to-1 segregation pattern. And you get all the possibilities. Mismatch deals with this randomly. So in fact, the possibility not shown here, that's just as equal, is that in fact it does fix it back to the original sequence. So you can actually get it back 2-to-2, red, blue, blue. Okay, but when it does not, then you get 3-to-1 segregation patterns, or you can get these alternate formation of spores, which you might detect if you look at neurospora, because we have an additional division and they're ordered. And so this is actually not a departure from Mendel, of course. It's just that you've essentially changed one chromatid sequence for the other because of a mismatch due to recombination. And yet you get a surprising segregation. Instead of 2-to-2, you get 3-to-1. That's due to the mismatch repair system that fixes these heteroduplexes. Okay. This process is called gene conversion, and it can involve single nucleotide polymorphism, as you can see here. Gene conversion can involve whole chromosome arms. Okay, so you don't have to convert a single nucleotide. You can even convert many alleles. You can actually change one locus for a deletion in that locus if it's converted by a chromatid that has a deletion there. Okay, so gene conversion is a result of heteroduplex formation during recombination, and because of branch migration, it can actually lead to exchange of huge chunks of chromosome arms. We talked about that. Summary slide. You need to remember these names. SPO11, the end of is meiosis specific. MRX XO1, these are responsible for the 5' end resection. I grant you the mechanism. RAD51 is strand invasion in mitotic cells, and DMC1 performs the function in meiotic cells. In meiosis, DMC1 is meiosis specific also, just as SPO11, but otherwise you have RAD51. And the book is kind of evasive. It says DMC1 plus RAD51. Well, DMC1 is the one in meiosis, and RAD51 is the one in non-meiotic cells. Oh, and REC8, please remember that one. That's the bacterial RAD51. So now, just a quick note, which we could talk for a much longer time. It's nice to have damage, but it needs to be detected. So all of these machineries that I talked about, UVR, ABC, NHEJ, recombination, and others that we haven't mentioned, they are elicited or activated by specific signaling and sensing mechanisms that will specifically sense the damage that is occurring and activate the cognate or the appropriate repair machinery. So here's an example. This is how double-stranded breaks are detected. And so double-strand breaks activate a kinase called ATM. And this kinase will then have substrates that are called checkpoint kinases. And they're checkpoint kinases because they are evolved in sensing problems and transducing that signal and into delaying or arresting the cells in G1. So when this is happening, the cells don't cycle. They stop before S phase. And they can only resume their proliferation if the DNA damage has been fixed. Okay, so there are two branches. There's the impact on the cell cycle and the pause or the delay or even the arrest in the cell cycle if the damage is great. That's one branch, and the other branch is the repair itself. So ATM, checkpoint kinases, and these kinases phosphorylate a molecule called P53. P53 is a transcription factor. So that is, it has a battery of genes that when P53 is activated, will be transcribed, that are normally not transcribed in cells that are not experiencing DNA damage. The phosphorylation of P53 allows it to liberate itself from another molecule called MDM2. So in normal cells that are not experiencing damage, MDM2 binds tightly to P53, and MDM2 is a class of molecule called an E3 ligase. And what you need to know is this is a class of molecules, MDM2, that lead to degradation of whatever they can bind to. And so in cells that are not experiencing damage, P53 is rapidly degraded. It never accumulates. Damage, as I just told you, leads to phosphorylation of P53. That phosphorylation makes that P53 no longer effectively binds to MDM2. It gets stabilized and is then able to bind to enhancers and induce transcription of a number of downstream genes. Let's talk about one of them, or two of them actually, but first of all, let's talk about this one called P21. P21 is a small molecule that is part of a class called CDK inhibitors. And P21 will bind to CDK cyclins. And I remind you, we talked about this when we talked about RBE2F and the G1S transition. And these cyclins, when activated, led to phosphorylation of RB, liberation of E2F, and we go into S phase. Well, in this particular case, P21 is the gene that's induced by P53. P21 is a P53 responsive gene, and P21 will then associate with CDK cyclins, more than just one type, and inhibit them. As a result, there is no phosphorylation of RB and cell cycle arrest specifically in G1. So while repair, as I just explained it to you, is taking place, the cells certainly don't go into S phase. This is a mechanism that has evolved to, in fact, allow cells to first fix the damage that they encounter before they can think about DNA replication and repair. If the damage is too extensive to be repaired, P53 can induce another molecule called BAX. That's why it's written here. This one on the right, and BAX then inhibit a molecule called BCL2, and BCL2 is an anti-apoptotic factor. So that is when we inhibit BCL2, that will push the cells into apoptosis and cell death. So P53 ATM orchestrates the repair and the cell cycle arrest. P53 mediates that signal, and if the damage is too extensive, then BAX is going to be the winning factor. It's also a P53 inducible gene, responsive gene, and BAX will lead to apoptosis and cell death. Questions about that? Okay. There are a number of syndromes in humans, and in fact, we have mouse models and models in other organisms that are linked to mutations and defects in these DNA repair systems. And so that is, these DNA repair systems in reality are not essential for life, but they're essential for the fitness of the cells, and that is that in their absence, there are a number of symptoms and or phenotypes that are linked to, in fact, disease. The main one is cancer. So mutations in the nucleotide excision repair system, this UBR-ADC that I told about in humans is called XP. And the XP proteins mediate that pathway. Mutations in XP lead to a disease that actually led to the name of these proteins. The disease is xeroderma pigmentosa. It should be here. There it is. That's defective excision repair. These patients are extremely sensitive to UV damage, and they have a huge increase in skin cancers. Another one that I want to point out is Ataxia Telangiectasia, and this is linked to mutations in fact, in ATM. This is also a syndrome that is linked to huge cancer incidence in these patients. So this underscores the importance of these pathways in health. Okay. So once again, the point is they're not essential for life. The cells can divide and even development can occur, but there's a huge price to pay if these systems are not functioning properly. There are other diseases that are, this is not even an exhaustive list. There are many, many diseases linked to defects in DNA repair systems. Questions before we move on to chromosomal aberrations? Yeah.
We're talking about
Yeah, so if the cells are not replicating, so in fact, most of our cells are not replicating, they're G0. I'll tell you, not much is known about that. It's not easy to study cells in G0. What I can tell you is that NHEJ actually works in G0 cells. So that's a system that's been documented to be active in G0. If the cell is one within, and I suppose the other DNA repair systems also are active. So we move on now to chapter 13. The important points here are now we're going to look at about point mutations and essentially fairly local um, mutations, right? But there can be rearrangement, chromosomal non-disjunction, and so errors and defects that are essentially affecting cells more globally. So the important points are non-disjunction in meiosis 102 can lead to aneuploidy, that is an extra chromosome or what's called a monosomy, which is lack of a specific chromosome. There are gross chromosome rearrangements that include duplications, inversions, translocations, and we'll go back to that notion that those DNA repair systems can generate those, such as an HEG recombination. So that can actually lead to problems in itself. And finally, transposons, we're going to talk about them and their mobile genetic elements that can actually lead to gene inactivation. So first of all, chromosome non-disjunction. This is something that, by the way, we've already discussed. The strengthened expression of, Calvin Bridges noticed that in flies. So you can look back at that. And you can have non-disjunctions of specific homologs in meiosis 1 or in meiosis 2. This is what happens in meiosis 1. So you have a non-disjunction here. That's a mistake. One of the products of meiosis 1 will lack this particular chromosome. The other one will have an extra one. And so we're talking about production of an individual that has a trisomy or a cell that has a trisomy, and another, if the other gamete gets fertilized, it's a monosomy. So that's an aneuploidy. Cells diploid minus one of the chromosomes. Remember one thing, if there's such an event here in the cell that has the extra chromosome, so what I want you to remember here is that when that kind of cell goes into meiosis 2 is in fact very simple. Chromosomes decondense, there's no DNA replication, they'll go back into prophase, and then each chromosome goes its own way. There's no more pairing of homologs there. So in a sense, this extra chromosome, if you look at the top of the slide here, is really not a problem in meiosis 2. Okay, it's dealt with, and nothing bad happens. It can just associate with the spindle just the same. And then you produce, in that case, two gametes that have just have one extra chromosome. We're just looking at that one chromosome here, and then a normal gamete fertilizing this oocyte, or it can be a sperm, in fact, lead to a trisomy. This can also happen in meiosis 2. In the case of a meiosis 2 event, then you produce two normal gametes because meiosis 1 is occurring normally, so there's no problem on one side of it. On the other side of it, this is the meiosis 2 non-disjunction, and you're producing once again trisomy and a monosomy. But in this case, you're producing two normal gametes in this particular. Um, so there are, as I'm sure you know, examples in humans of trisomy, monosomies. We know more about the sex chromosome aneuploidies because, in fact, they're not as uh, um, in, they're just as frequent, but they are not. They don't have such great consequences. So they are listed here. And in fact, of all the human chromosomes, we know only of three, trisomy 13, trisomy 18, and trisomy 21, because these are the only ones that, in fact, are tolerated in development, although they lead to really strong consequences, as you know. So, and in fact, the life expectancy for trisomy 13 or 18 is quite low. And I'd say, thankfully, now the life expectancy of the Down syndrome individuals has increased considerably due to progress in medicine. These individuals live productive life. That's a table that you see in old textbook. They tell you that for example, for trisomy 21, there's a strong correlation between the age of the mother and the frequency of that event. And that's true. It's very striking. And that's probably because oocytes are actually blocked in prophase 1 in mammals for many, many years, from in fact birth to puberty, and then there's only just one month that actually goes on. And so that arrest maybe is something that is kind of difficult to maintain as a person gets older. What they're forgetting to tell you is that uh, the age of the father of man also influences the genetic well-being of the child, but in a different way. That is, the rate of point mutations increases. Maybe I don't know if it's difficult to measure, so you don't compare the number, but the rate of mutations increases with the age of the father. Okay. Now, so we discussed how trisomy and monosomy can be produced during meiosis. But what happens to these individuals, and it doesn't have to be in humans, examples in plants and in animals, what happens when meiosis occurs in these individuals? And so what happens then is that in meiosis 1, this time we have a problem. I told you it was not a problem in meiosis 2, but now this trisomic individual or organism that in its germ cells is undergoing meiosis. So what happens is the homologs pair, and in fact the third chromosome will to some degree pair with the other two. So we're producing a trivalent chromosome, as shows, for example, the picture on the first slide of this set of slides. And the rules that we've discussed for meiosis still hold. That is, in metaphase, nothing happens until you have tension going from both poles. So the only way for that to happen is you have one chromosome or two that actually are linked to one of the poles in metaphase, and the other one or the other two, if it's only one, to the other. And so meiosis will occur only if you have any combination of two chromosomes going one way and one chromosome going the other way. Which they are doesn't matter. And so this is only one example, but essentially you can get gametes that are in fact normal that contain either the blue chromosome or the red chromosome or the pink chromosome and the other chromosomes, and the then what I'm trying to say here is that trisomic individuals actually do produce normal gametes to low frequency, but they do produce them. But the majority of them, of course, have an extra numeric chromosome. There's also non-disjunction that can occur in mitosis, and if that occurs in development, that leads to a degree of mosaicism in the individual. I think that's fairly easy to understand. So non-disjunction is not limited to meiotic cells. It can also occur in mitosis and mitotic cells. And now there are a set of things that are very interesting that I'm going to glide through. That's the question of polyploidy. So polyploidy is really, really fascinating from an evolutionary standpoint because it leads to the formation of new species. It probably occurs a bit more easily in plants because plants can self-fertilize. So a polyploid plant can create pollen and an oocyte that actually can have a set fixed number of chromosomes and form an organism, a viable organism. This can occur in different ways. Polyploidy can occur by multiple fertilization of a normal oocyte. It can occur by mitotic non-disjunction. And what we're talking about here is not a single chromosome. We're talking about what's called an endomitosis. That is, the spindle essentially completely collapses, and all the chromosomes then stay in the same area, and the nucleus reform. And this is what is essentially referred to as a mitotic non-destruction here. You can have essentially the same occurring in meiosis 1, forming essentially preventing the loss of ploidy in meiosis 1, and etc. So you can have different mechanisms that can lead to an increase in ploidy in an organism, say from a 2N organism, zygote, and resulting individual organisms 3N or 4N. I'm going to let you read through these things and how this can generate new species of plants. For example, wheat is next slide is a hexaploid compared to an ancestral species. There's actually a fitness advantage for species to acquire extra ploidy. This limits the problems linked to recessive mutations and such. So now I want to talk about chromosome rearrangements. And I've already sort of mentioned some of these things. The rearrangement shown here is called a translocation. This is actually, this can be a translocation. In this particular case, it's actually a deletion. So it's a break and the loss of terminal sequence of this chromosome, for example, human chromosome 5. This piece doesn't have a centromere. And so this creates what's called an acentric fragment. Centric because it doesn't have an acentric, it doesn't have a centromere. What happens then? They're lost. And why? I'm asking on this slide. Because they cannot interact with the spindle, and therefore they will not specifically go to one cell or the other. They may not even end up in the nucleus of the next cell, and that piece of DNA will be rapidly degraded. How does this occur? Through double-stranded breaks due to ionizing radiation or problems during replication that can create double-stranded breaks. And as I told you, this can be fixed, but if not, then the whole thing is lost. And these things can translocate to other chromosomes, in fact, as well, so they don't, when they translocate, they're not lost. And the activity responsible for this translocation will be non-homologous enjoining or recombination. You know how this works. There can be also massive deletions of chromosomes. They can occur internally in the chromosome. And so this can occur through two breaks. For example, there are deletions here. If the chromosome arm is broken in two parts, so two locations, then that piece can be lost and the terminal region can be religated to the rest of the chromatid by NHEJ. There's also recombination that can do this. And here's one example how this happens. It could be an unequal recombination between repeated sequences. So for example, this sequence B can recombine with the sequence A on the chromatid if they're repeated. And I told you you need some identity, but if it's there, if it's a repeated element, this can happen. And that leads to one product that has one of these repeat, and the other chromatid has 1, 2, 3. What you've done here, you've deleted a piece. So recombination unequal recombination can lead to extensive sequence loss. I would add something that is at the end of this segment here, that these elements that you showed here, these repeated elements, they look like things that are like they're just to make the point. It turns out there are many, many repeated elements in mammalian genomes. And these are remnants of transposable elements that were inactivated by mutations or reorganizations or massive deletions. But these elements are there, and they do constitute repeated sequences that can actually mediate recombination, as shown here. We'll talk about transposition at the end. Recombination can lead to deletion also by recombination within a single chromatid. Before I showed you two chromatids, so that was essentially in G2, right, after DNA replication. This can also occur in G1 with a single chromatid. And if you have a direct repeat, the sequence that can mediate recombination, you can actually recombine as shown here. And you go here, you have one element, and another one is something that's a circle that's excised if it's an intrachromatid. And this is lost, and you see you have deleted a piece, and this piece could be actually a huge piece of the chromosome. To something slightly different. Again, this is intrachromatid, and you follow the arrows here. Recombination between indirect repeats lead to inversions. So I'm showing you this, and this is, again, not theoretical. The elements that can mediate these events are present at significant numbers in genomes. Because we're going to talk about what happens when chromosomes do this, and there's going to be some important points following chromosomes that have inversions or deletions and things like that. So that's how they're created. I just wanted you to be aware of that. That's a slide showing the same thing. Deletion, indirect repeat, inversion. That's easy. Direct deletion, indirect inversion. Remember that. And another another type of cross-chromosome rearrangement, but the mechanism can be different. You can have two breaks, and you can have an inversion of that segment. Remember, an HEJ doesn't really care where the DNA is from. It doesn't know how the DNA used to be before the break. An HEJ just comes here, ligate whatever ends it sees, and you can generate with two breaks an inversion. And so you have two types of inversions, the types that involve the centromere, okay, and they're called pericentric inversions, and the ones that occur within a chromosome arm, not involving the centromere, and they're called paracentric inversions. If you think about it, in principle, pericentric and paracentric inversions, you'd say, well, that's really not much of a big deal, as long as all the genes are there. And you'd be correct. As long as the breakpoints don't affect gene expression, all the genes are there, it's fine. But these inversions have a huge impact on gametes that are produced by recombination. And so we're going to see what happens when, if you're in a line or in an individual that is heterozygous for these rearrangements, saying one chromosome is normal, the other one has an inversion, what happens to the gametes? I'm going to tell you right now, this is hugely important in genetics, because we're going to discuss how these notions have been used as tools in genetic analysis in blood.
If the genes were inverted, and let's say some of the alleles were linked, would they become unlinked?
Yeah, that's an excellent point. So I'm going to repeat the question. Let's say, for example, here, if you go back a slide, you see how, for example, A and B here are linked, right? And you shouldn't detect that by analysis of the progeny if you have the right cross. But then if you have an inversion, see they're farther apart. Now A and D are linked, and A and B are farther apart. And so in reality, yes, you'd see it by a higher frequency of recombinance. So they become less linked or even unlinked. It's a huge inversion. So before we go, I'm going to tell you something now. I'm going to repeat it when we see each other next week on Tuesday. This is what happens, for example, when you have a paracentric inversion. So you see this blue chromosome is normal, A, B, C, D, E, F, G, H, yeah? And this red chromosome is a paracentric inversion. A, D, C, B, E, F, G. So let's go through meiosis 1 and recombination. And so let's look at these chromatids. So we have this chromatid, for example, this blue chromatid here is involved in recombination. We have A, B, C, D, A. And this has two centromeres. You see that? A, B, C, D, A. It's right there. And the other chromatid, okay, is this one. It goes A, D, C, and then it goes A, B, C, D, A. See, I did it. It goes I, H, G, F, E. You go here, B, C, D, E, F, G, H, I. You've created a monster. You have no centromere, first of all. And this is essentially like something that's missing A, B, and C, and that has duplicated E through I, right? So what is important to see here is the inversion is completely lethal to the gametes that are recombinants. If you have a recombination event between the two non-sisters and one of them having an inversion, the recombinant gametes simply are non-functional. Okay? This one, because it's an acentric, it's lost, plus it's missing a lot of chromosomes, has a lot of duplication. This one has two kinetochores and actually is going to break randomly in the middle and give you something completely non-functional. So this is what I want you to remember for next week, when we go back here, is when you have inversions, okay, you will only get back functional gametes that are non-recombinant. And that is hugely beneficial to the geneticist and useful, because if you want to keep a mutation on a single chromosome arm, because you want to analyze it, you can use that kind of chromosome and you'll never have this mutation recombined. And you'll be able to trace it through crosses. So we'll go back to these points on Tuesday.